My name is Jim Sanford. I work with Dr. Gallo at UC San Diego, and I'm excited to talk today about a project that I've been working on for several years. The title of my talk is a little wordy, but inflammatory gene expression in keratinocytes is regulated by histone deacetylase 8 and histone deacetylase 9 and can be modulated by metabolites from the microbiome. So this is some work that we recently published last year in Science Immunology. I have no conflicts of interest to disclose today. Okay, I don't think I need to spend too much time stressing this to this audience, but the skin is an immunologically rich organ. There's a variety of cell types in both the epidermis and the dermis that participate in innate and adaptive immune processes, ranging all the way from the keratinocytes that make up the epidermis to cells like macrophages and monocytes and specialized T cell subsets within the dermis. But despite all of this rich immune activity in the skin, we are colonized by dense, abundant communities of microbes and typically reside without any sort of unwarranted inflammatory response to these communities. There are communities of bacteria, fungi, viruses that reside on the skin, within the skin, in the different appendages such as hair follicles. And as I mentioned, we typically live in peace with these bugs and there's no reaction, but there are some examples of inflammatory skin conditions where this homeostasis between host and microbiome has gone awry. And one of them is acne vulgaris. It's a very common inflammatory skin condition, affects millions of people every year with treatment costs exceeding the billions of dollars. And acne can be considered an inflammatory condition involving interactions between the host and these resident microbes, in particular one bacteria, Propionibacterium acnes, that likes to reside in the follicles. So in terms of thinking how we get from this healthy resting state of the follicle to these inflamed acne lesions, it's the same sort of bacteria that reside in the follicle, this Propionibacterium acnes. It has the same sort of structural ligands that might activate receptors on different cell types, such as toll-like receptors. But why do we typically exist, peacefully and then get to this shift to this sometimes very severe inflammatory condition? How do we get from A to B? A little bit more eloquently, the question is, does the skin environment trigger a microbial metabolic shift that leads to skin inflammation? So we started out with a hypothesis that when trapped in a hypoxic, lipid-rich, closed follicle, Propionibacterium acnes undergoes some metabolic changes and starts generating these bioactive metabolites that influence host cell types and only then trigger an inflammatory response from the skin. So we started off with some in vitro studies, culturing a strain of Propionibacterium acnes in the lab with and without a lipid substrate, glycerol, under low oxygen conditions. You can see visually using a phenol red pH indicator that when cultured with glycerol, P. acnes acidifies the media. You see this shift from the reddish orange color to yellow, indicating that it's using this glycerol substrate to make some acidic byproducts. If we take the supernatants from these cultures, use them to stimulate keratinocytes in vitro, look at inflammatory gene expression, we see that P. acnes by itself, in these gray bars in the middle, induces some level of inflammatory cytokine expression in genes like IL-6 and IL-8 and TNF-alpha. But when you take the supernatants from the strains grown with glycerol and add that to keratinocytes, you get a significantly higher induction of many of these inflammatory genes. If we analyze, that table got a little messed up. If we analyze the supernatants from these bacteria and look at what sort of short-chain fatty acids they produce, we see that P. acnes makes very high levels of propionic acid and valeric acid. And what do short-chain fatty acids do? One thing that they do is they inhibit interfere with host epigenetic processes. They inhibit the activity of histone deacetylase enzymes. So this graph shows that when we treat keratinocytes with propionate, butyrate, or valerate and look at global levels of histone acetylation, you see increased histone acetylation, indicating that HDAC activity has been inhibited. So to take a step back and sort of introduce histone acetylation, HDAC enzymes, how those all fit into this. In terms of packaging the large amount of genetic material into the small space of the nucleus, the DNA is wrapped around proteins called histones. These are folded into higher order structures, nucleosomes, chromatin, ultimately the dense visible chromosomes that we see in the cells. Histone proteins can be modified by the additions of various chemical groups. One modification is acetylation, so adding an acetyl group to lysine residues on histone proteins. And generally speaking, histone acetylation is associated with active gene transcription. It's thought that histone acetylation sort of loosens the interaction between DNA and histones and neighboring nucleosomes, resulting in a more loose, accessible state. And these acetyl marks can also serve as molecular docking points to recruit transcription factors and nuclear regulatory complexes. So the addition of histone acetylation is catalyzed by one set of enzymes called histone acetyltransferases, and the reverse side of that, the removal of these HDACs, or of the histone acetylation, is catalyzed by HDACs, histone deacetylases. And again, HDAC activity results in lower levels of acetylation, which is typically associated with a more compact, dense chromatin state and gene repression. So inhibiting HDAC activity pushes cells to a hyperacetylated state, which is typically associated with active gene transcription. But interestingly, in the world of inflammation and immune processes, HDAC inhibitors, which push things to a hyperacetylated state, have actually been shown to block inflammatory gene expression. They have an anti-inflammatory effect. This has been really well worked out in terms of the gut, where it's known that commensal microbes in the gut can break down dietary fibers and starches, generate high levels of short-chain fatty acids like butyrate, and it's thought that butyrate and these other short-chain fatty acids function to inhibit HDAC activity in resident cells like macrophages and dendritic cells, and dampen unwarranted inflammation, promote intestinal homeostasis. Indeed, when we culture monocytes in our lab, stimulate them with a toll-like receptor 2,6 ligand called MALP2, you can see the induction of a variety of pro-inflammatory genes from these monocytes with MALP2. But when co-cultured with butyrate and MALP2, the black bars on the very end, you see that the induction of these inflammatory cytokines is significantly decreased, inhibited. However, when we do these same experiments with keratinocytes, we see an exact opposite trend. So rather than an inhibitory, anti-inflammatory effect, short-chain fatty acids such as butyrate and propionate actually promote higher levels of cytokine expression from keratinocytes following toll-like receptor activation. We performed RNA-seq on keratinocytes, stimulated again with this TLR26 ligand MALP2. We looked at the sets of genes that were up and down regulated by MALP2 stimulation, and then evaluated the effects of co-culturing with short-chain fatty acids. And what you can see is that large portions of the genes from both sets are further enhanced by short-chain fatty acid-mediated HDAC inhibition. If you saw these same sorts of graphs for monocytes or macrophages, anywhere from 2/3 to 80% of the pie chart would be red, indicating an inhibitory function. So we've shown this pro-inflammatory effect of short-chain fatty acids on keratinocytes, but when you're talking about the follicular environment where P. acnes might be, where P. acnes metabolic products might be exposed to, there's a whole other set of cells, the sebocytes, that make up the sebaceous gland. So more recently, we've been investigating the effects of HDAC inhibition and short-chain fatty acid stimulation on sebocyte cytokine response. And much like keratinocytes, contrary to myeloid-derived immune cells, sebocytes show an enhanced TLR-mediated inflammatory gene expression when cultured with propionate, butyrate, or valerate. So looking at genes like IL-1 beta, TNF-alpha, TSLP, we can see that HDAC inhibition significantly enhances sebocyte expression of these cytokines. So up until now, we've been using these broad-acting HDAC inhibitory molecules, short-chain fatty acids, and we recently proposed or decided to investigate whether or not there are individual HDAC enzymes within the host cells that function to limit inflammatory gene expression. So within human cells, there's 11 classical HDACs. So the first thing we did was just run a panel of what is expressed in keratinocytes, identified a few sets of these HDACs that were more highly expressed than others, and went ahead and targeted those. So what we were doing was using siRNA-mediated gene silencing, looking if we can deplete individual HDACs. Does that phenocopy the effect of using broad-acting HDAC inhibitors? So to skip a bunch of negative data about ones that didn't work, we found out that when we deplete keratinocytes of histone deacetylase 8 or histone deacetylase 9, it looks just like the cells were treated with the short-chain fatty acids. MALP2-mediated cytokine induction is significantly enhanced when HDAC 8 or HDAC 9 is gone. This is true for many of the same sets of genes that are further enhanced by short-chain fatty acid stimulation. This is relevant if you look at staining of human skin. You can see that both HDAC8 and HDAC9 are highly expressed in epidermal keratinocytes, and importantly, they're expressed in follicular keratinocytes. And then again, thinking about not just the keratinocytes, but also the sebaceous gland and sebocytes that comprise the follicle, we do see that HDAC9 is highly expressed in cells of the sebaceous gland. And much like keratinocytes, if you knock HDAC8 or HDAC9 out of sebocytes, you see this same pro-inflammatory effect by measuring IL-1 beta, IL-8, CXCL10, the whole number of cytokines of your choice. So overall, what we've shown is that when grown in these hypoxic, lipid-rich environments, which is what we suspect a closed, occluded hair follicle is going to provide the bacteria, P. acnes can generate short-chain fatty acids that inhibit HDAC activity in host cells. When you inhibit HDAC activity in keratinocytes and sebocytes, this results in an enhanced cytokine response after TLR activation. And in particular, inhibition of histone deacetylase 8 or HDAC9 in these cells seems to be the key component. So we think that HDAC8 and HDAC9 typically function to repress cytokine expression, and when they're inhibited, that results in this hyperinflammatory response. So how we think this comes together in a new model of acne pathogenesis. We have a healthy resting follicle where there is P. acnes with all these toll-like receptor ligands, but it's not inducing any inflammatory response from the host epithelium. When there's an environmental shift, during puberty, when hormones are going out of control, skin is changing, sebaceous glands are hyperactive, follicles are plugging, we result in this environmental shift. We now have a lipid-rich, low-oxygen, plugged follicle that allows P. acnes to hyperproliferate, use the sebaceous products to generate short-chain fatty acids. These short-chain fatty acids in turn act on follicular cells like keratinocytes and sebocytes, promote the expression of pro-inflammatory mediators from these cell types, ultimately resulting in an immune infiltration and the marked inflammation that we see around clinical acne lesions. So with that, I'd like to thank Dr. Gallo and the members of the Gallo lab at UCSD, as well as everyone else in UCSD dermatology who's helped and supported me with this work. I'd like to thank the AARS for giving me the chance to speak here, and I'd like to thank a funding source, T32 training grant that we have. So thank you guys. I'm happy to take any questions now. This is my information in terms of where I'm going to be. I have a poster session and an e-poster session, so if you want to come nerd out and talk more data, I'm around, and I'll be here this afternoon as well. So thank you very
Jim,
much.
Thank you. I think we have time for 2 questions. Dr. Sabani?
Yeah, congratulations. A fantastic work. So how the HDAC inhibitor sends short fatty acid? Is it direct effect or something happens on pathway? So you have cultured keratinocyte with fatty acids that is a direct effect. I suppose that there's no contamination of TLR, so something should happen. It might be direct or not. So what's your thoughts?
Right. So there's a lot of data that's in our Science Immunology publication that I didn't want to dive into today, but we've investigated a few different pathways of how these short-chain fatty acids, whether or not they're actually inhibiting HDACs, which is a purely substrate binding pocket inhibition, or these short-chain fatty acids can activate surface free fatty acid receptors. They can stimulate downstream pro-inflammatory signaling pathways. So in our paper, we've shown that they don't activate these cell surface receptors. They don't influence P38, J and K, NF-kappa B activation, but they do alter overall chromatin state. So it opens up, adds acetyl groups in promoter regions of some of these cytokines. So yeah, there's a lot of data of things that it doesn't do, and we think it's all through histone acetylation. Direct binding of the short-chain fatty acids in the active site of the HDACs, correct.
Okay, one more question.
Hi, I'm Jason Meyer from UCSF. Nice talk, thank you. In thinking about where these the substrates for creating these short-chain fatty acids come from, I know there's a lot of interest, like back in the 70s, in blocking bacterial lipases that might, release free fatty acids and glycerol. And correct me if I'm wrong, but it was, I think, really surprising that they didn't, even though they could really suppress the hydrolysis of the triglycerides, the Propionibacterium's numbers were not reduced and the acne wasn't reduced. So just wondering if you have any thoughts about that, where the substrates are coming from, or,
Yeah, that's a great question. So, I think the substrates are coming from the sebaceous glands, so their sebum products, and I think a combination of host factors and bacterial lipases can degrade these into smaller bits that the P. acnes can utilize for these fermentation pathways. So yeah, that older approach of blocking bacterial lipases. Alternatively, if you can block the hypersebaciousness, that seems to give better outcomes.
Thank you. Thank you.
Thanks.